Alright, welcome back. We're going to jump into section 6.6 here and take a look at the cytoskeleton and the network of fibers that are involved in organizing structures and a number of different activities within the cell. And this is composed of three main types of molecular structures. We're going to talk a little bit about microtubules. We're going to talk a little bit about microfilaments. And then we're going to talk about the intermediate filaments and the role that all three of them play in getting stuff accomplished inside the cell. The picture that you're looking at is one that's been fluoresced, and once you do that, you reveal this very complex structure of tubules and fibers that you would not normally know exist within the cell. So the role, again, is primarily support. The cell, if we're talking about plant cells, they have a cell wall. That's really all the support they need. It's a very rigid structure, and that's going to hold everything in place. But if you're talking about an animal cell, that's a whole different ball game. The cell membrane is very flexible. It bends and distorts very easily when it comes in contact with a force. So there needs to be some type of internal network of girders, so to speak, that are going to give this cell its shape and support and keep it from collapsing. There's a very important role played by the cytoskeleton in terms of moving things around within the cell. And this is some of the really cool stuff that we're going to watch and take a look at with the video clip on the inner life of the cell. They show those motor proteins moving stuff around on these girders of the cytoskeleton, or monorails as they talk about it here in the PowerPoint. Here is a transmission electron microscopic view of a microtubule that has a motor protein on it, and this vesicle is being moved somewhere throughout the cell, and it's being moved by just the action of these little legs that are motor proteins powered by ATP. There are three main types of fibers that make up the cytoskeleton. Again, microtubules, microfilaments, and the intermediate filaments. The thickest of the three are the microtubules. These act as like the girders of the cell. These are the primary structure that is keeping the cell a certain shape, keeping it together so it doesn't collapse. Remember, different types of cells are going to have different shapes that are specific to the function of that cell. A nerve cell looks very different from a muscle cell because it's doing a different job. And so the microtubules are involved in giving that shape, and you'll see some pictures of that as we come up here on the next slide or two. Microfilaments are oftentimes called actin filaments. These are the thinnest components of the cytoskeleton. Here we're talking about very, very thin protein fibers that are probably going to be wound around one another, braided, and that's going to give them some strength, but they're very thin, and their function would be movement. Specifically, if you're talking about muscle cells, their ability to contract or expand depends totally on these microfilaments. The intermediate filaments then are somewhere in between. They're in that middle range. So this is a good table to take a look at in your book, table 6.1, because it really is going to break everything down that I'm talking about over the next few slides in terms of their job, what the structure is. For microtubules, they are hollow tubes. The thickness, I don't expect you to remember the thickness other than to recognize that it's thicker than the other two that we're talking about. And maintenance of cell shape, keeping that cell from being compressed or squashed, so to speak, is going to be a very, very important function of this type of tubule. You also have a very important function when the cell gets ready to divide. It's going to take the chromosomes and divide them up. That's called mitosis or meiosis, and we'll get into that a little bit later on. But moving those chromosomes around from one end of the cell to the other is done by the microtubules. Cell motility, the construction of a flagella, cilia, That is going to be done with microtubules. And finally, if we need to move an organelle around, say we need some mitochondria in a different part of the cell, it'll be the microtubules that'll be primarily involved in taking that organelle and dragging it to some other location within the cell. Microfilaments, again, two intertwined strands of actin, which is a type of protein, stranded around one another or braided around one another like I was talking about. You're primarily going to find this when you're talking about muscle contraction, and we'll show you some pictures of that coming up. Cytoplasmic streaming, what the heck is that? Well, the amoeba organism, which we looked at under a prepared slide, not a living slide, those little finger-like projections that are coming off of the amoeba is the end result of microfilaments hurting the cytoplasm and kind of pushing it out against the cell membrane in a certain direction. So when the amoeba wants to crawl along the bottom of the pond, the lake, whatever, it is using cytoplasmic streaming to do that. And then they have a small role in cell division also, mitosis, meiosis, and its cleavage formation. That's something we'll talk more about when we get to the chapters on mitosis and meiosis. Finally, the intermediate filaments. These are, again, proteins that are supercoiled into thicker cables. You can see a pretty good picture of an intermediate filament right here. And that intermediate filament shows this tightly bound cable of protein fibers. And they're going to be used, again, in the maintenance of cell shape. That's with all three filament. Specific function with the intermediate filaments is to get the nucleus anchored, and maybe some other organelles like, say, the endoplasmic reticulum, get them anchored in one location of the cell so they're not sloshing around and moving around and making it difficult for the cell to get to the nucleus, get in there and copy a specific gene to do, various functions that it needs to do. So again, these next few slides just kind of reiterate what I've talked about on that table 6.1. So we got, again, microtubules, their job, their function, we covered that. Centrioles we didn't talk a lot about, and centrioles are involved in moving the chromosomes around. We did mention the chromosomes being separated out during the process of mitosis. It is the centrosome and the centrioles that are creating this kind of network of cables that are going to get all the chromosomes lined up in the cell so that we don't accidentally send a few extras to one side and not enough to the other. So we get them all organized, spread out. We can see, okay, we got to send this one here, we got to send this one here. And in animal cells, this is a very complicated process where the centrosome uses these centrioles, each with 9 triplets of microtubules arranged in a ring, usually around another pair of microtubules. In this case, with the centrosome, it's just a ring structure. You can see a cross section of a centriole that has been taken by a transmission electron microscope. Cilia and flagella, you probably know what cilia and flagella are, but let me kind of talk a little bit about these structures because they are incredibly important in the world of a one-celled organism. Oftentimes these organisms are going to need a way to move around in their environment, and that oftentimes is done with either 1 or 2 or 3 long extensions of the cell called flagella. This obviously is a sperm cell, and this very long whip-like tail allows for the sperm cell to swim towards its destination, which would, of course, be an egg cell. Cilia are these much smaller, more numerous hair-like projections that are on the outer surface of the cellular membrane, and they oftentimes will beat in unison like oars off the side of a boat. And the cilia and flagella, they do have one thing very much in common, and that is this motor protein called dynin. This protein is what is primarily involved in driving the bending movements of the cilia or the flagella. It's the trigger that is pulling that cilia down to one side or that flagella to one side. All right, in this picture you can see the dynein proteins. They are located between the pairs of microtubules that we were talking about, 9 pairs, oftentimes arranged around 2 central pairs. And it's this connection here that is pulling those microtubules in one direction or another. And they're trying to demonstrate that here a little bit more. See, here it's a little easier to see how the proteins are pulling one end down or the other end down, which then in turn causes this cilia to be bent in one direction. And of course, we're using ATP to do this. One of the many functions of ATP in the cell is to cause the cytoskeleton to move, in this case the cilia or the flagella. The microfilaments, what were some of the main things that we talked about that you're going to need to know? Well, these are primarily, again, structural. Their job is to deal with tension as far as the pulling forces within the cell. This girder network that we've set up inside the cell to keep its shape is being constantly abused by outer forces. In your intestines you have a series of projections called microvilli, and their job is to absorb food. All these little needle-like, finger-like projections allow for the intake of food molecules, water, etc., because you've really added to the surface area by stacking them end on end on end. And the microfilaments are what is holding all this mess together, so to speak. And the actin fibers that are attaching the microfilaments to the intermediate filaments are going to be flexible enough to allow for contraction, to cause these microvilli to move back and forth so that we can get more food molecules in contact with the membrane, the plasma membrane, so we can start bringing some of those molecules into the digestive system. So these microfilaments then, they also have a function in cellular motility. This protein called myosin, in addition to actin, are working in unison. The myosin and the actin will slide one over the top of one another when the cell contracts, like say a muscle cell, and then they'll slide back out when it expands. So it's this constant contraction expansion that is allowing these fibers, pulling these fibers over the top of one another. And here's a pretty good picture showing the myosin fibers and the actin fibers sliding over the top of one another. Here again, the actin is this braided protein, the myosin is much thicker, and the scanning electron microscopic view of it really demonstrates how they're sliding over the top of one another every time the cell needs to contract to move bone. Same thing is at play here. We're using these subunits, these actin subunits, to contract and to get cytoplasm pushed in one direction, causing the extending pseudopodium, which is the name that we give these finger-like projections that are commonly associated with amoeboid type organisms. And then finally, when we get the LOD and we start looking at that underneath the microscope, one of the things you're going to notice, especially when you have the lamp turned on to view the cells, that warmth, that light, the plant's going to respond to that. It's going to want to do photosynthesis, and you're going to see the chloroplasts start to move around inside the cell, and it's because of this cytoplasmic streaming that is getting these chloroplasts moving around so that they can take in light and they can carry out the process of photosynthesis. Here you can see on the screen an example of what I'm talking about with cytoplasmic streaming. Look at the chloroplasts moving around. This is a series of Elodea plant cells that are exposed to light, and so they've been kicked into action in terms of photosynthesis. All right, finally, intermediate filaments. Again, main function, support cell shape, we talked about that. But I think the more interesting and probably more relevant function is this need to get certain organelles locked into the cell so they're not floating all over the place. And the number one organelle that we're talking about here is the nucleus.